Good morning. This is the chapter Plant Tissue Culture for the fourth semester B.Pharm students. That is coming under the third chapter or third unit in your syllabus. Plant Tissue Culture. In today's class, we are going to see about the introductory part, that is the definition, the history, and what are the requirements for the plant tissue culture. So this is what exactly, if you take the plant tissue culture, it contains the definition part, the historical development, the types of the various tissue culture methods, the nutritional requirements, growth and their maintenance, and at last the applications of plant tissue culture in pharmacognosy. That is nothing but in how it is useful in the developing a whole intact plant, or otherwise the secondary metabolites which are used industrially as well as in the medicinal field. And when you just see the tissue culture, that is nothing but, what do you mean by culturing? That is maintaining the growth. So tissue means what? It is the group of cells which has the similar characters or growth characteristics. Okay. So growing of the cells which are having the similar characters in an artificial medium, apart from the natural growth, is called as your tissue culture. That is crudely. And this tissue culture may be divided into two main areas, that is plant tissue culture or animal tissue culture. That means in plant tissue culture, the culture is obtained from the plants, and in case of your animal tissue culture, from the animal cell. Here we are concerned about the plant tissue culture. When you just come to the definition of this plant tissue culture, it is an in vitro method of cultivation where the plant cell or a tissue is kept under the aseptic and controlled environmental condition in a liquid or a semi-solid nutrient medium for the production of primary and secondary metabolites, or to generate into a whole intact plant. Okay, that is what you're going to do over here. Apart from the natural conditions which is provided for the growth of the plant, you are isolating a plant single cell or a tissue, that is nothing but the group of cells from the plant, and you are going to grow that particular isolated cell or a tissue under controlled environmental conditions in the aseptic area, and where you are going to provide the nutrient as a liquid medium or as a semi-solid medium. Thereby you are going to achieve the regeneration of the intact whole plant, or you are going to produce the primary or secondary metabolites which are produced by that particular plant. Okay, that means you are growing the cell or a tissue in a controlled system in the aseptic area by providing all the conditions like your temperature, your nutrient and everything, and you're regenerating it into a intact whole plant, or as well as you are going to produce the active ingredients produced by them. In other words, you can say that it is an experimental technique in which the mass of cells, that is otherwise called as the callus, that is nothing but it is a mass of undifferentiated cells, are produced from the explant tissue. Before going into that, you should know what do you mean by an explant. Explant is nothing but it is a small part of a plant. That is, from the part of the plant, a small area has been removed and it is sterilized, and that is used for developing the whole intact plant or for the production of the secondary metabolite. That is called as your explant. And this callus is produced in the process where it can utilize directly or regenerate the intact whole plant, or otherwise from the growth we can extract the active medicaments synthesized in that, or otherwise what you can do, you can manipulate or you can change the production of your primary and secondary metabolites. The change in the production of primary and secondary metabolites, in other way it is called as biotransformation. What do you mean by that biotransformation? That is nothing but in an usual plant cell, the biosynthetic pathway is going on. That means how the secondary metabolite or a primary metabolite is produced from the starting material. Okay, so here what you are going to do, whichever the way you want to change the product, you will be inserting the precursor so that your desired primary metabolite or a secondary metabolite can be brought about at the end. Okay, so this when you we will discuss in the later chapters or in the later upcoming classes. That is about the biotransformation. Okay, fine. And this plant tissue culture, it refers to the cultivation of a plant cell. Okay, that is from an isolated individual cell to a multicellular tissue. Okay, do you understand? We are isolating a single cell and we are allowing that single cell to grow in an artificial medium with all the wanted environmental conditions, everything you are providing artificially, and you are allowing that single cell to multiply and to differentiate into different types of tissue, and finally you are going to get the whole intact plant. Okay, and this particular tissue is grown on the agar medium. It produces a mass of undifferentiated cells. Okay, that is called as your callus. And this callus culture is convenient for starting and maintaining the cell lines, and also to study the various things, and also to bring about the organogenesis and meristem cultures, and to have a detailed experimental studies on these kind of techniques. Okay, and this in vitro cultivation of plant cells or organs is when you do that, usually it come across with some problems. That means these problems can be uh, avoided by uh, providing the aseptic conditions. So what are the problem you're going to encounter during this plant tissue culture or when you're growing the plant cell under the in vitro condition? The very first thing is the attack by the microbes. Why the microbes will attack? Because in the preparation of nutrient medium, you are using the glucose or the carbon source, the nitrogen source, everything where there is a chance for the growth of the microorganisms. Okay, so that is why you are doing all the things under the aseptic techniques that to keep away the microorganisms from the plant cells. Okay, and next one is when the plant tissue culture is going on, what happens? First the cell has to be multiplied and then it has to be differentiated into a particular organs. Okay, so at that time, for the growth as well as for the differentiation of the cells, you have to provide a suitable nutrient media. And apart from providing the suitable nutrient media, you are also you are also required to produce the exact environmental conditions where it is going to grow naturally. Okay, so the providing these conditions are little difficult, and by taking the stringent rules and other procedures following in a stringent way, you definitely you can provide a suitable nutrient media as well as the other environmental conditions. Okay, so these are the main two problems what the plant tissue culture technique will face. That is, one is major is the attack from the microbes, another one is providing of the selected media, and also the exact environmental condition which is required for its growth. And next, when we see the advantages of this plant tissue culture, the first thing is the availability of raw material. Okay, and that means what? The raw material which is required for the plant industry for the production of the phytopharmaceuticals. That means the availability of raw material. So when you're doing the or when you're getting the raw material from the cultivated plants, sometimes you may not able to get this supply which is required for the industry in a exact quantity what they require. Why? Because when you're cultivating the plant, due to the climatic conditions or due to the other environmental condition which affects the growth of the plant, may not be able to supply the regular and uniform supply of the raw material. So that is why when you are producing the secondary metabolite through the plant tissue culture, you will not face any kind of these problems. That is the growth of the plant suppression, or otherwise the production of secondary metabolite is suppressed by the other environmental conditions. Why? Because you are providing everything in a very good calculated manner in an artificial medium, so that there is no suppression of the production of secondary metabolite. So whatever the quantity or how much ever the quantity the industries are required to produce the phytopharmaceuticals, that can be supplied. And the second advantage is the fluctuations in supplies and qualities. That is again due to the climatic changes on or the other kind of environmental conditions, or when the crop is affected by the disease during the cultivation, or due to the some of the seasons. Okay, the production of secondary metabolite in the plant may vary, but that kind of problem we cannot face in the plant tissue culture when you're producing the secondary metabolite. And next one is the next advantage is the new methods for the isolation. So whatever the thing novel isolation methods you have, you can also do on the experimentation, where the new methods can be employed for the isolation of the components produced by plant tissue culture, and for which the patent also can be made. And next one is the biotransformation. That's what I was talking to you before. There's nothing but, each and every secondary metabolite is produced in the cell by the biosynthetic pathways. Okay, so each and every step in the biosynthetic pathway has its own metabolite and the other things. So here what you are going to do, the any compound what you are going that is which is involved in the biosynthetic pathway can be slightly modified, and that can be introduced in the plant tissue culture, so that you will get whichever the product you are desiring. Okay, that means already you know the structure of a secondary metabolite. So by slightly modifying the functional groups, you can alter the pharmacological activity of the group, activity of the active medicament. Okay, so if you want to alter the pharmacological activity of the medicament, slightly the functional group has to be changed. So that can be brought about by this biotransformation techniques. Okay, and next advantage is the disease-free or the desired propagule. Well, what do you mean by that propagule? It is nothing but it is a part or any kind of thing which is used as an explant to produce the whole intact plant or to produce the secondary metabolite. Okay, that is called as your propagule, right? So this disease-free and desired propagule can be obtained by this tissue culture method. That means when you are producing the plant or the secondary metabolite by the plant tissue culture method, the desired propagule can be stored and maintained for any period of time without any damage during the transportation or sometimes during the subsequent plantations. Okay, the next one is the biosynthetic pathway. Uh, that's what I told you. The secondary metabolite is produced in the cell or a plant cell from the starting material, and to the last product you have many kind of synthesis or the different steps involved in that. And uh, that study can be done by this plant tissue culture. That is nothing but the tracing of the biosynthetic pathway. Tracing means what? Finding out the various steps involved in the biosynthesis of active constituent right from the precursor. What do you mean by precursor? That is the starting material. Understand? And next one is the next advantage is your immobilization of cells. What do you mean by immobilization? Mobile means moving. Immobile means you are not allowing the cell to move. That means how you will not allow the cell to move. You will make your cell to get entangled in a matrix. Okay, or you are anchoring the cell, thereby you are reducing the movement of the cell here and there in a nutrient medium. So when you are doing like this, what are all the advantages? That means you can avoid the loss of the cell, you can avoid the loss of the enzyme, so you can stabilize your cell or an enzyme for a longer time. The stability can be maintained for a longer time. Okay, so this immobilization makes the cells, that is it will preserve the cell as well as it facilitates the transportation and biotransformation. Okay, so this immobilization of cells also you will be seeing it in detail in the later classes. And next you can have the continuous and uniform biomass is obtained, so that there is no prevention in the supply of the demand. And the medicinally important compounds can be synthesized, and which is not having the specific procedures to synthesize chemically. And the useful natural compounds can be obtained by this method without the normal conditions like soil and change in climate and other things. And also you can improve the medicinal plant species by doing some of the genetic engineering techniques during the plant tissue culture. And propagation of plant without seed is also can be controlled, and in a defined way that also can be done. Okay, so these are all the different advantages of this plant tissue culture. Okay, so right from the beginning, if you see the availability of the raw material to the industries without any obstruction, and the fluctuation in the supplies and qualities can be maintained, and you can develop the new methods for the isolation in the biotransformation. It is very highly it is useful, and you can produce the disease-free and desired propagule, and also you can trace the biosynthetic pathways, and it is also helpful in immobilization of the cells and production of secondary metabolites through that. And the continuous uniform biomass can be obtained by this, and for the compounds where the chemically cannot be synthesized can be prepared by this method. And also the useful natural compounds can be obtained without any obstructions due to the soil conditions or climatic conditions. And also you can improve the medicinal plant species, and also the propagation without the seed. That is nothing but the vegetative propagation also can be done in a defined and controlled manner. Okay, so that is about the advantages of plant tissue culture. And next you can see the disadvantages. You can see that is the high level of expertise is required. Yes, you have to have a very profound knowledge over there. And if there is any small error, that may lead to the complete collapse of the product or the plant, because the whole batch has to be substituted and it has to be started newly. And lots of chemicals are required to maintain the different conditions of the uh, during the growth of plant tissue culture, as well as to maintain the high purity. Okay, and again there is no chances for mutation over here. And again the culture is made on the artificial medium. So sometimes if you are not carelessly, if you are not maintaining the culture medium, naturally sometimes it may lead to the depression of unusual metabolic pathways, okay, which is not beneficial to the biotechnologist. And in the some of the cases, uh, the secondary metabolite production is negligible. And the protocols for individual plant differs from uh, the each individual. And change in the medium, especially the environmental conditions and other things, so everything you have to take into account. And to maximize the cell mass or the callus or the suspension, uh, when it becomes very much dense, right? And it may produce some kind of problems that usually the number of cells increases, there will be a chances of decreased amount of nutrient medium to each and every cell, and aeration will not be there. Okay, and so that the plant may grow in a, sorry, the different cells will grow in a different way. So that everything has to be taken into account very carefully. And sometimes the instability, and sometimes the growth may be very slow. And apart from that, cost wise it is highly expensive. And above the all, you have to maintain the aseptic conditions. That is, you have to maintain each and everything, each and every procedure, the area, the instruments what you are using, everything should be uh, free from the microorganisms. So which is a very great challenge and very great task to the biotechnologist. Okay, so these are all the disadvantages of the tissue culture. So but when you just monitor all these things in a very careful way, definitely you can wonders in this area of experimentation. And when you just see about the historical development of this plant tissue culture, this principles of plant tissue culture was involved during the year from 1838 to 1839. And in that period, it was, the theory was advanced by Shildon and Schwann. And later for some years it has been left as such. And in the early 19th centuries, the studies over this and various developments has been started. In the year 1902, Haberland, he proposed the concept of in vitro cell culture. And in 1904, Hanning, he cultured the embryos. Embryos, you know very well, it is a part of a seed. From that, the cell explant is taken, and then they have developed the cruciferous species. And in the year 1922, Holt and Robbins, they cultured the root tips and stem tips. That means the explant is, or the isolated cell is taken from the root or stem. And in 1926, the very first plant growth hormone, that is auxin, indole 3 acetic acid, was uh, isolated when they were doing the studies in plant tissue culture. And in 1934, White, he is a person who introduced vitamin B as a growth supplement in plant tissue media, especially when they are doing the tomato root tip culture. And in 1939, Guthrie, White, and Nober, they established the callus cell culture. And in 1941, the overbeak was first added to the, that is in the medium they can add coconut milk for the cell division, to induce cell division in datura. And in 1946, Ball, he raised a whole plant of lupinus species by shoot tip culture. That is, from the shoot of the plant, the explant was isolated, and from that explant the whole intact plant is raised. And in the year 1954, Muir, he was the first person to break the callus into a single cell. That means a single cell has been isolated from the callus tissue culture. What is a callus? It is an undifferentiated mass of cells. Okay. And in the 1957, Skoog and Miller, they gave the concept that the hormones, auxins and cytokinins, can be employed to initiate and to undergo the cell division and organ formation. And in the year 1959, Rinnett and Stewart regenerated the callus clumps from embryos. And then also they converted the callus into the suspension cultures, that is from carrot, Dacus carota. And in 1960, Cocking, he was the first person to isolate a protoplast from the cell by enzymatic degradation. And again in 1960, Bergman, he is the person who filtered the cell suspension and isolated a single cell by plating techniques. Okay, in your microbiology, you might be studied about your plating technique. And in 1962, Morashik and Skoog, they have developed the nutrient medium with the high salt concentration. Again in 1962, Kanta and Maheshwari, they have developed the fertilization technique in the plant tissue culture. And in 1966, Stewart demonstrated the totipotency of regenerating carrot plants from single cell of tomato. What do you mean by totipotency? It is an ability of a single cell to regenerate into a intact whole plant when it is subjected to grow on the nutrient medium which supplies all the nutrients under controlled environmental condition. That is called as totipotency. It is a capacity of an individual cell to develop into a whole intact plant in a artificial medium under controlled conditions. Okay. And these are all the various historical developments in the field of plant tissue culture. And next we have to see about the basic requirements of the plant tissue culture. So what are the things you require when you are going to do the in vitro plant tissue culture? The very first thing you require is your plant material, that is nothing but your explant, equipment and glasswares, and you have to maintain the aseptic condition, and the requirements for your washing and storage facilities, and the media. You have to require a place for each and every procedure to follow. Okay, so there you have a media preparation room, sterilization room, nutrient room, and transfer room, culture room or incubators, and proper and optimum aeration you have to supply during the plant tissue culture, and well equipped observation or recording area. Okay, so these are all the basic requirements for the plant tissue culture, and now we will see one by one what is what. First one is the plant material. The plant material, that means nothing but the explant. It should be disease free and it should not be too old. Okay, and then uh, the particular species or the variety or the genotype what you're going to use, it should be of the right one. And then the in vitro germinated seedlings, chosen as seed, everything has to be sterilized. Understand? And then the plants should be healthy, and whenever the stage we are taking it to use as an explant, it should be healthy, uh, and it should have its own totipotency. Okay. And maybe, what do you mean by an explant? Maybe you may take an internode, or sometimes you may take a part of a expanded leaf, or sometimes from the root or from the fruit, whatever the part of the plant you are taking. It should be healthy, and it should have its highest totipotent nature. Okay. And the equipments and glasswares you require is, you have to need a incubating chamber, that is nothing but the laminar airflow cabinet with a UV fitting light. You might have seen in your microbiology lab, where all the aseptic techniques or transfers has to be done. And then incubator, which should have the temperature control, and usual recommended temperature for the moist heat. Most of the tissue culture is 36 degrees centigrade. And also you should require an autoclave for sterilizing the glasswares and the media, that is the moist heat sterilization, whereas for the dry heat sterilization, that is sterilization of the glasswares and other things, there sometimes you might be using the hot air oven, that is nothing but your dry heat sterilization. And you need refrigerators and freezers for the storage of reagents, tissue culture stock solution, chemicals, etc. And you should also have the microscope to observe the cell characteristics. The microscopes may be of simple or special microscopes with the camera fittings. Okay, so take the photographs and to do the things. And the stage should be properly large enough to accommodate all the large roller bottles in the specific cases. And also you require the pH meter for adjusting the pH of the medium. You require the burner, spirit burner or a gas micro burner for the sterilization of the instruments or other heating purposes. And next is the washing up equipments. The washing facilities of glasswares, pipettes, and in usually they are done by the soaking these apparatus in deep baths or washing sinks of the stainless steel or propylene, polypropylene, or should be there or provided to do the manual washing and rinsing. As I already told you, hot air oven should be there for the dry sterilization of glassware, and standard siphon type pipette washers should be there, and which is during the washing it should be soaked in the detergent for overnight, and that can be done. And washed pipettes should be rinsed with the deionized water, and it should be dried. And also the stainless steel pipette dryer also should be there, and water purifier to get a pure and deionized water, and centrifuge to increase the concentration of the cell suspension, and shakers, okay, during the growth, or the when the growth is allowed slowly, the nutrient media has to be shaken well to distribute the nutrients and other conditions in a proper way, and a balance to weigh the various nutrient materials for the, that is the chemicals which is used to prepare the nutrient medium, and proper shells to store all the things, and the other smaller surgical instruments like your scissors, scalpels, and forceps for the extant preparations and their sterilizations, and culture vessels usually made up of borosilicate glass, which includes the test tubes, conical flask, bottles, special flat tubes, etc. And all these glassware should be of specific dimensions. Okay, usually 100 ml conical flask or test tubes with 25 to 150 size. And the other glasswares like the measuring cylinders, beakers, funnels, petri dish, graduated pipette, conical flasks, etc., which is required for the various measurements during the preparation of nutrient medium. And apart from that, the miscellaneous things required may be your non-absorbent cotton plugs, screw caps, or polyurethane foam, which is required to close the mouth of the culture vessel. And again, the aluminum foil over the cotton plug or a screw cap, the aluminum foil has to be kept and it has to be closed properly. And also you require some labels, marking pencils, and hand lens, plastic disposable syringes, bottles, hot plates, stirrer, etc. And apart from that, you also require a microwave oven and to melt the solidified media for the purpose of pouring into the culture vessels. Okay, and next condition what you have to maintain is your aseptic condition. That is the plant materials, or that is nothing but your explants or the equipments or culture media or the room. Always it should be free from the microorganisms. Usually dry heat or wet heat or ultrafiltration or chemicals can be used for the sterilization process. And when you're doing the sterilization, that is especially in the sterilization of explant, the surface sterilization is very, very important. That means the explant may be taken from different parts of the plant, that may be from the seed or a fruit or a stem or leaf. And when it is extracted from different parts of the plant, it has to be sterilized. When you are doing the sterilization, you cannot subject the live cells into the moist heat or dry heat. Naturally, it has to be sterilized by using the agents with the appropriate concentration. Okay, so here I had put down some of the examples in their specific concentrations. That is 9 to 10% of calcium hypochlorite for about 5 to 13 minutes. That means after taking the explant from a part of the plant, it has to be soaked in the 9 to 10% calcium hypochlorite solution for about 5 to 30 minutes. And second one may be 2% sodium hypochlorite solution. That is about for 5 to 30 minutes again. And wherever you're preparing the solution, you have to use a double distilled water after sterilizing the double distilled water. That water has to be used to dissolve these substances. And again, 10 to 12% of hydrogen peroxide. The time specified is 5 to 15 minutes. And 1 to 2% of bromine for 2 to 10 minutes. And 1% solution of chlorine water or mercury chloride or silver nitrate or any antibiotics also can be used. Sometimes the absolute alcohol is used for the hard tissues. That is for the sterilization of the explant. And for the sterilization of the other equipments, maybe the dry heat method, that is hot air oven for about 1 hour at 160 degrees centigrade. And sterilization of equipments can also be done by washing with some of the chemicals like chromic acid, sulfuric acid mixture, or otherwise hydrochloric acid, strong nitric acid, or detergent solution, alcohol. Usually the incubators and autoclaves are cleaned or sterilized by this method. And another method of sterilization is moist heat or wet heat sterilization, where the glass waves and culture media are sterilized by using an autoclave. The sterilizing condition maintained is the temperature at 121 degrees centigrade at 15 pounds per square inch pressure for 15 minutes. And sometimes for the thermolabile components, the liquid media can be sterilized by ultrafiltration techniques. Sometimes antibiotics are added to the medium to prevent the growth of the microorganisms. Here some examples has been given, that is potassium benzyl penicillin, streptomycin sulfate, gentamicin, etc. And some of the chemicals like alcohol are used to sterilize the working area or the instruments. And so the sterilization of explant is over. sterilization of the materials, culture media, everything is over. And if you are maintaining the area where you are performing these experiments without sterilization, it is of no use. Okay, the area also should be sterilized by fumigation method. Everything, the preparation room or inoculation chamber or laminar airflow or whatever it is, they have to be used by this fumigation techniques. Okay, and next one is the washing and storage facilities. For washing, always the fresh water supply should be there and disposal of water, wastewater should be there. And there should be a space for the distillation unit where the double distilled water has to be prepared and it has to be deionized. And the working tables, sinks, wash basins to wash the apparatus and equipments should be there. And sufficient space is required for placing the hot air oven, the washing machines, pipette washers, and other equipments. And for the storage of dried glasswares, a separate dust-proof cupboards and the cabinets should be provided. And next one is the media preparation room. The media preparation room should be spacious enough to accommodate all the labwares, culture vessels, equipments, chemicals. And apart from these things, it also should be well equipped with a refrigerator, freezer, and the specific places for storing this refrigerator and freezers to store the media and the stock solution. And next one is your sterilization room. So it is nothing but it is a tissue culture lab. It is usually desirable to have a separate sterilization room for sterilizing all the things, maybe your culture media, the glasswares or metallic equipments like scissors, cal. Okay, so usually I already spoken to you regarding this. The sterilization can be done by the autoclave, that is moist heat, or by the hot air oven, that is dry heat. Okay, and next one is your nutrient medium, that is the media where the explant has to grow, should have all kinds of requirements for its normal growth and functions. So the nutrient medium, it is composed of the inorganic nutrients, which includes the macronutrients like nitrogen, phosphor, potassium and calcium, and micronutrients like boron, copper, iron, and manganese, zinc. And apart from the inorganic nutrients, it also requires the organic nutrients to supply the carbon source and other things. And also it requires the vitamins like B1, B6, B3, B5, etc. And also it requires the amino acids like arginine, asparagine, cysteine, hydrochloride, cysteine hydrochloride, glutamine, and carbon source is usually provided from glucose or maltose, and also the presence of growth hormones like auxin, cytokinins, and gibberellins, abscisic acid, and ethylene to provide the cell initiation, cell division, and cell proliferation. And other media substances like protein hydrosylates, like yeast extract, and fruit extracts, especially the banana extract, coconut milk, and solidifying agents like agar, alginate, gelatin, etc. And usually the iron source also should be there, and also some EDTA, that is a chelating agent, and antibiotics, which prevents the growth of the microbes. And once if you weigh all these nutrients in a required proportion according to the different media available, and it has to be dissolved in a double distilled sterilized water, deionized sterilized water, and then the pH of the preparation has to be adjusted. The pH range should be around 5.6 to 6 before autoclaving. Okay, and once the sterilization is over, the other procedures can be followed. And next one is the transfer room. This is nothing but where the laminar airflow hood is present, where all the transfer of the nutrient medium into the culture vessels or transfer of the explant, everything has to be done in an aseptic manner under the laminar airflow with the help of the UV light. Okay, and when the culture replantation or transfer or any kind of step has to be done in a clean aseptic area under laminar airflow. Okay, and the culture room or the incubators, once the transfer has been done, it has to be provided with the exact temperature for its growth. So that can be provided by the use of this incubators. The specific condition of temperature, humidity, air circulation, everything, light also provided. And the incubation chamber or area, it should have both the light and the temperature control device, which is should be which should be managed round the clock. Okay, over the 24 hours of period. And usually the light can be supplied by the fluorescent lights. That is, it should be white in color, and it should be cool, and it should have the high output. So if it is subjected that the culture should be kept in the darkness, it has to be kept without the light. And the temperature range we have to maintain is at 25 degrees centigrade plus or minus 2 degrees centigrade. And next one is the proper and optimum aeration. Apart from providing the exact light and temperature, you are also supposed to provide the aeration. Adequate aeration is required for the cell to grow. That is, you know, already in your microbiology, you might have studied about the aerobic and anaerobic organisms. Okay, so wherever the aeration is required, we have to produce the optimum aeration. And if the cells are to be cultured on the semi-solid media, they do not require any kind of aeration. Why? Because the cells are present in the surface of the semi-solid media where aeration will be there. But when the tissues are grown in the liquid medium, that is nothing but the culture in case of suspension, there the aeration is required. So in that condition, the special devices should be kept over there to provide proper and optimum aeration. Okay, so these are all the various requirements, the basic requirements for the plant tissue culture to run. Okay. So this is about the very general introduction, that is definition, its characteristic advantages and disadvantages, and the history of the plant tissue culture, and the basic requirements required to run a plant tissue culture lab. And in the next classes, we will see about the other aspects, that is the different types and how it is done. Thank you.